Hello students, I am Dr. M. Bhushanam, Assistant Professor in Zoology, Maharani Science College for Women, Bengaluru. Hope all is well. Take care of your good health during this pandemic time. Today, we shall learn a topic in zoology related to paper 7, Developmental Biology and Organic Evolution, that is 3.1, Neo-Darwinism, under which we shall learn the concept of gene pool, gene frequency, Hardy-Weinberg law, its definition and significance. The objective of the present study is to understand the genetic composition of a population and the forces that determines and changes its composition. The contents includes, under Neo-Darwinism, the concept of gene pool, gene frequency, Hardy-Weinberg law, its definition and significance. Students, before we go in detail about the topic, you please remember the concept of gene pool and gene frequency is important for short note questions, and Hardy-Weinberg law, its definition and significance is important both for 3 marks and 5 marks. Coming on to the introduction. Students, any noticeable change that occurs gradually in the characters of organisms, which is transmissible or inheritable from the parents to offsprings in due course of time, is called organic evolution. In simple, it refers to the gradual changes that are inheritable. Fine. How does these changes takes place and what are the reasons for it? Where are the proofs for these changes that had taken place all through these millions and millions of years? No one has seen, but scientists have put forth various theories to explain these gradual modifications in due course of time. These theories come under the theories of organic evolution. There are historical and modern theories to explain these changes, like theory of special creation, then Oparin hypothesis, theory of Lamarckism, theory of Darwinism, etc. Today, the most accepted theory includes modern synthetic theory of evolution or Neo-Darwinism. Here, the concept of germplasm and its relation in natural selection together brings in a certain of slow and heritable changes. It was given by Weizmann, and the germplasm present in the gametes of an organism or a species is responsible for the inheritance of characters. Otherwise, all heritable changes or variations starts from the reproductive germ cells of an organism. Based on the theory of germplasm given by Weismann, many other scientists also put forth their ideas in the form of postulates, which are collectively called as modern synthetic theory of evolution or Neo-Darwinism. The scientists of modern synthetic theory includes biologists like Dobzhansky, Haldane, Fisher, Sewallwright, Stebbins, Ernst Mayr, etc. The major theory of modern synthetic theory includes two important postulates. Number one, gene variations or genetic variations. In simple, it says changes that occurs in genes. Number two is natural selection, which otherwise includes fittest individuals are selected by the natural forces or factors. Students, here, the gene changes or genetic changes when we concentrate upon, it includes mutations, recombinations, hybridization, genetic drift, etc. Mutations are any sudden inheritable changes of a gene. As we all know, genes are the functional units of chromosomes, or they are the regions that includes nitrogenous base sequences which codes for proteins. Any change in the nitrogenous base sequences of a gene changes or alters the protein what it codes for or what it synthesizes. Thus, the changes may be related to either structure of the chromosome or number of the chromosome. The structural changes includes the deletions, duplications, translocations, inversions, point or shift mutations, etc., where changes are related to the structural part of the chromosome or the gene. Whereas the numerical changes of chromosomes includes change in the number of chromosomes related to a particular cell of a species. Here, addition or deletion of whole chromosomes either in single number or in their sets takes place. Accordingly, we call them as aneuploidy and euploidy conditions. I think in last semesters you might have learnt about trisomy, nullisomy, polyploidy, euploidy, etc. These are all the examples related to the numerical chromosomal aberrations. Then next comes the recombination. It is a process where the genetic changes takes place during the crossing over phenomenon of meiosis. Then comes hybridization process, which is another process of genetic change that we see during the phenomenon of offspring formation. The next we have is the genetic drift, which relates to the elimination of genes from a group of individuals or a population. So all these changes are related to the genes. So this is the major basis for the modern synthetic theory concept. The second important postulate of the theory is natural selection. It is the Darwinian postulate, right? It is popularly known as theory of natural selection, according to which the fittest individual is selected by the environment or nature for the survival of a particular species. So, fittest individuals with the genetic variations together corresponds to the concept of modern synthetic theory, where we find individuals with new characters will slowly, in due course of time, enters into the concept of change of characters in them. So, the concept of Neo-Darwinism is related to these two important postulates. One is genetic variation, number two, natural selection. These two has to together act and should help in the formation of new species. So this theory defines the concept of evolution in a very different way. Modern synthetic theory can be defined as the changes that occur in allele frequency within a genetic population. So modern synthetic theory, as we have understood, it is the combination of two postulates. Number one, genetic variations it should show. Number two, the natural selection it should show. So combination of genetic variation, natural selection is modern synthetic theory of evolution. So it defines the concept of evolution as changes that occurs in the allele frequency within a population, or it simply says it is the genetics of evolution. In our words, to understand it, when changes are more, new species are getting formed, and this changes in new species is transmitted to the generations through the process of interbreeding. So this is what exactly the concept of modern synthetic theory otherwise says. Changes that happens in genes are related to the alleles. We know allele is the alternative form of a gene. So gene is represented in the form of alleles. These alleles will show changes in them. That is what we call it as genetic variation here. So these changes has to be selected by the nature, and that will form a new species in future if the changes are transferred or transmitted to the generations. So we call it as inheritance. So inheritance of the genetic variations that happens in alleles is exactly what we call it as the process of evolution. So, the study of frequency of genes. Frequency is a common term. Students, remember, frequency refers to number of times it appears. What is referring to? Frequency of genes. So, it otherwise refers to number of times a gene or genotype. Genotype also refers to representation of gene in the form of alleles. So, frequency of a gene and genotype that appears in a population is called as population genetics. So, in simple, we call it as study of frequency of genes and genotypes in Mendelian population is called as population genetics. Appearance of genes and genotypes many number of times in a given Mendelian population, and its study we call it as population genetics. So, the organic evolution relates to a concept called as modern synthetic theory, which is nothing but Neo-Darwinism, which says that there should occur some gene changes, and these changes has to be selected by the nature or environment. We call it as natural selection. Such characters need to be transmitted or inherited to the generations. So such type of inheritance, we call it as genetic evolution. So the concept of genetic evolution relates to another important concept referred as population genetics, which deals with the study of frequency of genes and genotypes in the given Mendelian population. So how many times these genes and genotypes will appear in the given genetic population is referred as population genetics. When we say Mendelian population, it has two features. The individuals of population should mate randomly. So these are conditional concepts. So when I say Mendelian population, it refers to the population which mates randomly. So by chance, so chance or randomness of mating is selected. So particular individual can mate with any of the individuals of that particular population. So that random picking is allowed. That is called random mating. Number two, the next is all genotypes of individuals have equal survivability. So otherwise I can say no specific genotype is selective. Here down towards the right side we have a picture depicting peppered mud Biston butyllaria. Here the color, size, environment are not the criteria for the mating of this particular species. We have many species. So mating is random. Any organism can mate with any organisms of the any individuals of the population. Number two, genotypes are also not the barriers. So we have random mating amongst the individuals of a population, and all genotypes are randomly picked up. So specificity of genotypes are not selected. So such type of criterion is what exactly we find in a Mendelian population. So this condition of having no restriction for mating among intermating individuals is generally called as a panmixis, and the population is called as panmictic population. A population where there is no restriction for mating of individuals of the intermating population. We call it as panmictic population. An advantage of random mating population or panmictic population is gain of changes or variations, and this changes also relates to the adaptability. When an individual of a particular population mates its selected individuals of this group, what happens here is that variations are transmissible. So change in genes are possible, and these changes makes those organisms to easily adapt to the environment in which they are. This is advantage of random mating. Number two, the gene pool concept, that is random mating of individuals, occurs in the same species and same gene pool is maintained. In simple, I can say when the individuals of two different geographical areas mates, the genetic variations are much same. So thereby what happens is that if those changes are accepted by the nature or environment, they are transmitted. So such changes we call it as genetic variations, and these variations are if are selected by nature for the fittest individuals, then it is an advantage for the organic evolution. So what happens to the gene number there? Gene number totally changes because new genes are getting added up to the original individual population. Then what is this gene pool referring to? It is the sum total of genes present in the given Mendelian population. So total number of genes present in the given population, we call it as gene pool. Students, we have understood two important concepts here. Number one, random mating population or panmictic population. Number two, gene pool concept. What is gene pool? Pool refers to sum total of gene is referring to gene pool for a Mendelian population. For example, if there are 100 individuals in a given population, let us assume each individual has, let us consider 1,000 unique genes or alleles in them. So there are 100 individuals in a population. Each individual contain 1,000 genes in it. So what is the total number of genes? 100 into 1,000. That is equal to 1 lakh. So this 1 lakh number of genes which are unique of that population, we call it as gene pool. So gene pool refers to the sum total of genes present in the Mendelian population. Students, remember, gene pool is a gene reservoir. Okay, it is a reservoir of all genes present in that Mendelian population. So here is another example. To understand what gene pool is. So definition of gene pool is, it is the sum total of genes or unique alleles, that is specific genes present in a given Mendelian population. It is the reservoir of all genes or alleles of a population. Example here, when we look at a population includes individuals of A, B, C, D, E, 5 individuals. Students, we will understand what is the gene pool again. So there are 5 individuals in a population, let us assume. There is a unique P gene or allele present in A, B, D and E population. E individuals. So it is absent otherwise in C. So we have altogether 5 individuals, but P allele is present in 4 individuals, that is A, B, D, E. It is absent in C. So it is present in 4 individuals. So what is the gene pool percentage of P gene? We have total number is 5, but unique allele P is present in only 4. So 4 by 5 is equal to 80%. So the 80% of P allele is the gene pool of P. Are we able to understand? Are you able to understand? So P allele frequency is 4 by 5, that is 80%. After we understanding the concept of gene pool, now we will understand the types. There are 3 types of gene pools. Number 1, primary gene pool. Secondary gene pool. Number 3, tertiary gene pool. Here, the quality of interbreeding and offspring formed matters a lot to us. So this is the basis for the classification of gene pool. In primary gene pool, the unique alleles occupy the same locus of 2 individuals. Mean to say 2 individuals that involves in their mating or inbreeding shows the location of the gene common, so same loci, such that the inbreeding rate success is so high that it produces fertile offsprings. So when two individuals carrying the unique alleles, when they start mating, they show the formation of successful offspring, which is fertile individual. So fertile offspring, if it is obtained, we call it as primary gene pool. The next one is secondary gene pool. In secondary gene pool type, the loci of the unique alleles, that is the location where the unique alleles also remain same, but the interbreeding rate is very less, leading to the formation of sterile offspring. Students, remember, if two populations with the unique alleles mate, if it produces fertile offspring, we call it as primary gene pool. If two populations with unique alleles occupying the same locus, if they inbreed, and if the inbreeding rate is lesser, forming a sterile offspring, we call it as secondary gene pool. In tertiary gene pool, the loci of two individuals that involve in the process of mating for a unique allele remains same, but no intermating or no inbreeding takes place. Thereby, no offspring is formed. So we call the condition as tertiary gene pool. So condition number one is primary gene pool, where the inbreeding individuals will produce fertile offspring. Secondary gene pool, where sterile offsprings are formed. Whereas tertiary gene pool, no offsprings at all are formed in the case. The next concept under population genetics that we need to understand is gene frequency. It is also important for the short notes. Gene frequency refers to the number of times it represents, it appears. Gene frequency or genetic frequency or allelic frequency all refers to the same concept here. It refers generally how many times a gene or an allele is repeated and how often a given gene appears in a given population. So if you have a particular gene, how often is that gene appears in the group of the population? This we call it as gene frequency or genetic frequency, can also be called as allelic frequency. So simply it refers to the proportion of an allele or a gene present at a particular locus in a random mating population. Okay. Let us take an example to understand gene frequency. If we have 100 plants of 4 o'clock plants called Mirabilis jalapa, an example for the incomplete dominance. Down in this slide we can see red is dominant, white is recessive. When both are crossed, we get an intermediate pink color flower. And when two pink color flowers are crossed, we get back the original parents as well as the FN hybrid appearance in it. So of 100 plants, 25 are red, 50 are pink flowers, 25 are white flowers. We know to calculate the frequency. When one red, one white flower, homozygous dominant, homozygous recessive flowers, when are crossed, we get heterozygous intermediate pink color flowers. This is F1 generation. when two F1 hybrids, that is two pink color flowers, when are crossed, we tend to find the F2 generation showing the ratio of 1:2:1, where one is red, the other one is white, the middle two refers to the heterozygous pink color. In simple, I can say, if there are 100 flowers, 25 flowers are red, 25 flowers are white, and rest 50 flowers are pink. So to calculate the allelic frequency of dominant allele capital R or uppercase R in 100 flowers, the total number of R alleles divided by total number of alleles that are specific locus is the allelic or genic frequency of R allele. The total number of R allele, capital R allele, divided by total number of alleles. We have seen what is the total number of R alleles present in 100 individuals. We know of the 100, 25% are capital R individuals. So 25 out of 100 has this R alleles, capital R alleles in simple. So here in the slide, when we look at 25 individuals of capital R are there out of 100. So 25 into 2 is 50. Correct. So we have twice capital R here. So what is the number of capital R alleles? Twice 25 numbers. So that is 50. But there are also a capital R alleles in pink flowers. How many pink flowers are there? 50. So only one capital R is present in them. So total number of R alleles, when we take account, 25 individuals will have one capital R and another capital R. R and R, capital R and capital R. So total number of alleles in 25 number is 50. This is in red flowers. We do have one more variety called pink flowers of 50 number. There only one capital R is there. So they are also 50. So 50 plus 50 includes 100. So 100 is the total number of capital R alleles. What is the total number of alleles? What is the total number of alleles? We have totally 200 number of alleles. How it is possible? You will have to go back and see. Total number of capital R alleles are 100. Total number of small r alleles also 100. So 100 plus 100, capital R and small r together is 200. So allelic frequency of capital R allele is equal to total number of capital R alleles divided by total number of alleles present. So 100 by 200 is equal to 50. So allelic frequency of capital R allele is 0.5. That is 50% otherwise. Similarly, of small allele also will be 50%. That is 0.5. So when we look for the total allelic frequency of capital R and small r, 0.5 plus 0.5 is equal to 1. If we give the representation of allelic frequency of capital R of red flowers equal to 0.5 and small r is equal to 0.5. If I could represent capital R frequency of allele as small p and represent the allelic frequency of dominant allele of capital R is small p and recessive of small q. So lowercase p, lowercase case q. Then the sum total of, that is p plus q, of allelic frequencies of dominant and recessive is always equal to 1. In simple to say, when I total up, sum up allelic frequency of capital R and small r, that will be equal to 0.5 plus 0.5 is equal to 1. So P plus Q concept is equal to 1. So here this total allelic frequency of dominant allele in a Mendelian population and recessive allele in a Mendelian population, when we select, it will be equal to the sum total 1. This is one concept. The next comes the concept of genotype frequency. Okay, gene frequency, genetic frequency, allelic frequency refers to one concept is what we have understood now. The next is genotype frequency. Genotype represents the representation of genetic trait. For example, you have learnt in your previous semester, homozygous dominant character is represented as capital T, capital T. Heterozygous tallness is represented as capital T, small t. And dwarfness as small t, small t. So this representation of the genetic characters, a genotype appearance, is exactly referred as genotypes. Now, the genotype frequency refers to how many times the given genotype appears in the given population, or it is the ratio of appearance of genotypes in the given Mendelian population. So how many number of times a given genotype appears is called genotype frequency for a Mendelian population. It is also called as zygotic frequency. So to understand it with an example again. Genotype frequency of red, pink and white flowers. Let us consider again of the 100 number, 100 plants. 25 are red flowers with the genotype capital R, capital R. 25 are white flowers with small r, small r. Rest 50% are pink flowers. And the frequency, when we look at 25 by 100 for capital R, the capital R genotype is 0.25. Heterozygous capital R, that is pink flowers, capital R, small r is 0.5. And again, white flower, small r is 0.25. So, the sum total of genotypic frequencies, when I add up, when I sum up, of the genotype capital R, small r and small r, we know that alleles representation for one capital R, that is dominant allele, is small p, and of recessive allele is small q. So if that is the case, the capital R can be represented as small p. Capital R, small r, heterozygous is represented as PQ, and small r, small is QQ. Here, the sum total of all these genotype frequencies, when we look at 0.25 plus 0.5 plus 0.25, that is also equal to 1. If we could give the frequency of genotype, capital R, capital P, capital R as small p, capital R, small r is P and Q and small r as Q and Q. The sum total of these also, that is PP plus PQ plus QQ is equal to 0.25 plus 0.5 plus 0.25, that is equal to 1. So sum total of the genotype frequency also remains equal to 1. Sum total of genotypic frequency also remains equal to 1. It was Hardy, an English mathematician, and Weinberg, a German physician, in 1908 gave together the concept of Hardy-Weinberg law or Hardy-Weinberg principle. It is a principle used in population genetics. Students, remember, this is a 5 marks question, important, most repeated question in the question papers related to organic evolution and population genetics. Now let us understand first the statement of Hardy-Weinberg law. It states that in a large Mendelian population where random mating, that is panmictic population, they are random mating, where any individuals can select any individuals for the mating. So random mating population, the sum of frequency of alleles and sum of frequency of genotypes, allelic frequency sum and the sum of genotypic frequency, both remains constant or equal to 1. Condition is if there is no selection, no mutation, no migration, no genetic drift. If there are no external forces that acts on this randomly mating population, if there are no disturbance factors for this random mating population, then in such large group of population, the sum total of genotypic frequency and genic frequency or allelic frequency remains equal to a constant 1. So this is what the law says. Students, this law is very important for the examination point of view. Just now we have learned the sum of allelic frequency and genotypic frequency of a given population remain constant, equal to 1. If there are no action of all these forces, this law is mathematically represented by using the formula, mathematical formula, by the Hardy-Weinberg scientists. Let us now understand it. The mathematical representation of the law for the same example of Mirabilis jalapa, when we take up, we have dominant character is red alleles, capital R, capital R. In heterozygous condition, they form pink flowers. And homozygous recessive condition, small r, it forms white flowers. Where small p is the allele frequency of a dominant allele Small q is the allelic frequency of a recessive allele. Of the 100 number, allelic frequency of P, when we look at it, is 0.5 as what we have learned in previous slide. Q is also 0.5. So P plus Q is equal to 1. Now, if we know the allelic frequency of one particular allele, let us say P if we know, then we can also easily calculate Q value. Q is equal to 1 minus P. If we know the value of Q, we can easily find out the value of P, 1 minus Q. So P plus Q is equal to 1. The genotypic frequency of the same alleles when we represent, it is PP plus 2PQ plus QQ. It represents the algebraic formula. It can be summed up as P square plus 2PQ plus Q square, which is possible when P plus Q whole square algebraic formula, when we expand, it becomes P square plus 2PQ plus Q square. That is also equal to 1. Just now we have understood. Whether it is P plus Q is equal to 1 or P plus Q whole square is equal to 1, both remains common. That's what it says. Condition here is there is no external forces of selection, mutation, migration and genetic drift acting upon this population. Students, I think the concept is clear. Okay, P plus Q is equal to 1. It represents allelic frequency. Okay, sum total of allelic frequency. So, sum total of genotypic frequency is PP plus 2PQ plus QQ. That is equal to 1. That represents genotypic frequency. So, both remains equal to unity or constant. That is equilibrium. So, the Hardy-Weinberg law hence is also called as Hardy-Weinberg equilibrium principle, because it is equivalent to constant 1. Here is a slide to understand Hardy-Weinberg principle again. So, sum total of allelic and genotypic frequencies of a random mating population remains constant if there is no influence of evolutionary forces as what we have discussed just now. Conditions here are population should be larger, there should be no mutation, no migration, no natural selection, no selection of sexes and no genetic drift. Small p is the frequency of dominant allele, small q is the frequency of recessive allele. So, sum total of small p and q, that is equal to allelic frequency, is equal to 1. Similarly, the random mating individuals will also show presence of genotypic frequencies p square plus 2pq plus q square is also equal to 1. And down is the example to represent the Hardy-Weinberg principle again. So, random mating in a population is needed very much for the Hardy-Weinberg equilibrium, along with no external forces. Here is an example in this slide to explain so. If we add up any of these forces, mutation, selection, migration, genetic drift, etc., to this population, if I give specificity for a particular A individual of a population should mate only B, if this conditional clause is given, we call it a selection process. Or if I bring in certain mutational changes in the population, then what happens is this frequency will not remain equal to 1. The value varies. So, that's what is the importance of random mating. The factors that affects gene frequency and genotypic frequency is selection. Selection simply refers to natural selection, a process which favors the survival and reproduction of the fittest individual in the given population. We also select by human efforts, we call it as artificial selection. Students, in this slide, when we look at, we have the original population of insects with green and brown color. Due to predators, the green color ones are eaten away by the bird there, and thereby certain changes, the brown ones are getting inherited within. The coloration is the background on which they remain matters a lot, so that it confuses the predator. Both the organism and its background both remains of the same color, so that the green ones are selected here. So thereby certain changes is brought within the brown organisms here, and that forms a new species in future. So this is how selection occurs. If this selection process is same, the green ones from the original group will slowly reduce. But it is not the case. They should remain equivalent to that frequency constant condition. But if there is a selection, there is a disturbance between allelic frequency and genotypic frequency remaining equal to 1. Similarly is the fitness. When we look at here, the relative reproductive success of different genotypes of a population in the same environment under natural selection is called fitness. Simple words to explain it is, the most adaptive individuals with the high reproductive success are selected by the nature. So it is capital W is the fitness representation. Always when the value of W is equal to 1, then 100% of survival is seen. When the fitness value is 0, the genotypes totally is lost. So always the value ranges between 0 and 1 here. Next is selection coefficient process. It's a process which represents the rate of elimination of different genotypes from a given population. Otherwise, we can understand that. Just now in the previous slides, we have understood that the green varieties are eliminated from the individuals by the nature by using predators. So this is called selection coefficient concept. So it is represented as capital S. Students, remember, it is also called as selective disadvantage. If the value is greater, lesser the survival rate. If the S value is lesser, the survival rate is more. So it is a disadvantage for the animal actually. Then comes the concepts like mutation. So selection should not act upon the group. Next, it should not, the process of mutation should not occur in the group of Mendelian population in Hardy-Weinberg group, principle to appear. Mutation generally refers to sudden heritable change in the organism. It is the DNA that undergoes a sudden change in the nitrogenous base. So gene itself will change. So genetic variability is seen. So you can see here 2 green species of insects all of a sudden forms a brown individual. This is what we call it as a mutation. When this kind of changes occurs, the gene allelic frequency and genotypic frequency will never remain constant. Then comes the concept of migration. Migration refers to movement of alleles from one population to another population. We also call it as gene flow. Whether it is movement of immigration or emigration, that is incoming or outgoing allele population, let it be of, both are harmful, because both will, if are occurring in a given Mendelian panmictic population, a random mating population, what happens is that the allelic frequency and genotypic frequency never remain equal to 1 because there is an imbalance. Similarly, the another important concept which acts as a natural force on the Mendelian population is genetic drift. So genetic drift or random drift refers to random change in gene frequency due to sampling error, selection error. This generally happens in a very small group of population. When we look at the picture above, in the first jar we find green and brown color balls. So it is represented 60% of them are brown ones and 40 are green ones. As the time goes on, 80 brown ones and 20 green ones are seen. Further, you find 100% brown ones and 0% green ones. Furthermore, the 100% brown ones are seen, no green ones. So what's happening to those green balls within the jar? We are selecting them to reduce the population of those green balls, and indirectly we are making that brown balls to remain increased in their number. So here the genes, each of the color of the balls represents the genes. Here the gene frequency of one particular allele, whether it is dominant or recessive, whatever the condition be, it is slowly getting drifted away, slowly getting departed away, moves away. So the other one is getting selected by the nature. So this particular condition we call it as genetic drift. Students, remember, in Hardy-Weinberg principle, these forces of migration, mutation, genetic drift, all this should not act on, but that doesn't happen at all. That condition doesn't survive in nature. So in natural population, Hardy-Weinberg principle, to maintain that equilibrium is very difficult because environment has to interact with all these evolutionary forces. Thereby, slowly the Hardy-Weinberg principle will have its changes. So this is about the importance of different factors that acts upon Hardy-Weinberg principle. The genetic drift, what we have understood just now, there is drifting of gene frequency. It can occur in two types. Number one, founder effect, and number two, bottleneck effect. Both the conditions will alter the gene frequency in the next generations. So totally a new population will be the product of the or result of the effect. Founder effect. Founder means something to find new. So a new generation of our population is seen. So that's what we have seen here in the previous slide. So totally of green and brown gene alleles, what we have found is totally leading to brown allele genes. So something like that, a new population gets exposed from a main population itself. This is what we call it as founder effect. So there are many reasons for this, and this concept we are going to understand in the next classes to come. The significance of population genetics, when we look at, where we have learnt about gene frequency, genotype frequency, Hardy-Weinberg principle, gene pool, and different evolutionary forces that acts upon the given population. So all this ideology or understanding of these terms we have learnt. Next is, we also have learned that the study of gene frequency in a population will help us to know how a natural evolution process would have taken place in the environment. And we also find that the Hardy-Weinberg principle to maintain its equilibrium is so very difficult because of the external forces of mutation, genetic drift, migration, etc. And the same knowledge can be applied on the development of composite and synthetic varieties and pollinated crops. The scope of population genetics helps us to understand how the frequency of alleles and genotypes change with the period of time between the populations, how new characters gets imbibed by mutations, and how this genetic variations or changes slowly gets inherited to the generations of offsprings, and how a new species gets formed in the environment. So this is about the importance of population genetics. Well, students, after we understanding the concept of population genetics under Neo-Darwinism, now we shall look into the MCQs. So variations of genes, alternative forms of genes, is nothing but alleles. So answer is A. Second question. For a recessive trait to appear, the individuals must receive the genes from both the parents of father and mother. So answer is A, true. Question 3. Individuals who possess a copy of both dominant and recessive allele in them, we call it as heterozygous condition. So answer is D. Question 4. What is genetic drift? It is the gene frequency that will change over time because of the random mating in a small population. So answer is B. Question 5. Imagine you raise goldfish as a pet dealer, and you have 10,000 fish in a large tank, but due to an electric problem, 95% of fish perish overnight. The remaining 5% are left to breed and to repopulate. So that is what is forming a new population, passing their genes, traits to their future generation. So what type of genetic drift it is? It is answer A, bottleneck event. Bottleneck event refers to the smaller population which is left after the selection process that will be continuing into a newer generation. So whatever is left out with a small number, that will continue a new generation. So it's a bottleneck event. A gene locus has two alleles, capital A and small a. The frequency of dominant allele is 0.4. Okay, allele. That is small p. Small p is 0.4. The frequency of homozygous dominant, heterozygous, and homozygous recessive individual. So when I say homozygous, heterozygous, it represents the genotype frequency. So P into P is P square. So it should be 0.4 into 0.4, 0.16 for capital A, capital A. So D is not selected. A, B, C can be selected as for the first one is concerned. And we know Q is 1 minus 0.4. So that is equal to 0.6. So 0.6 into 0.6 is small a, small a. So 36 is the answer. So even C is ruled out. Now A and P is left. Of the A and B, we have 2PQ, 2 into P into Q, 2 into 0.4 into 0.6, that is 0.48. So answer is A. What does P square below mentioned Hardy-Weinberg principle equation indicate? So Hardy-Weinberg law is P plus Q, that is allelic frequency is equal to genotypic frequency. That is equal to P square plus 2PQ plus Q square. That is equal to 1. That is Hardy-Weinberg law. So what does P square here it indicates? They are asking. So it is the individuals with homozygous dominant. So answer is C. This condition is essential for a population to be in Hardy-Weinberg equilibrium. So to have the Hardy-Weinberg equilibrium, these are the factors that are required. We require random mating. Yes, definitely yes. No mutation. Yes. It remains in a large population. Yes. So answer is D, all of these. Ninth question. The statement that describes Hardy-Weinberg law the best. So correct statement of Hardy-Weinberg law otherwise. It is impossible to predict the expected allele frequency mathematically. No. In large population, dominant allele becomes more prevalent. No. Allele frequency changes over a period of time in a large population. That is also no, because it's drift. And D is mechanism of inheritance in a large population does not change allele frequency. So answer is D. So allele frequency should remain constant. If it is changing, we cannot call it as Hardy-Weinberg law. This is true of the population which are included in Hardy-Weinberg equilibrium. Entities migrate constantly. No. Population should be limited and small. No. So large population, no migration is done. No selection also. So C is the right answer. Mating is random. So after we understanding the MCQs, now let us look at the outcome of the present session. Hope you all have learnt about the genetic composition of a population, different forces that determines and changes the Hardy-Weinberg equilibrium. The references includes the web references of Wikipedia and Britannica.com, and book references includes Organic Evolution by Veerbala Rastogi and Cell Biology, Genetics, Molecular Biology and Evolution and Ecology by Verma and Agarwal. Thank you all students. If you have any doubts related to this first unit part, please post your doubts to my personal WhatsApp number. Thank you all. Thank you all.